day. This is the Professor, CBMD PhD, and today's Friday, September 8th, 2023. It is 2:11 and 29 seconds, AM Eastern Standard Time, and today's research, tumor growth. So let's jump to it here. As super enhancer supercharges pancreatic tumor growth, researchers identified new set of molecules that drive growth of human pancreatic cancer cell lines, potentially leading to new therapeutic targets. Date September 6, 2023. Source: Salk Institute. As scientists identified new set of molecules that drive the growth of human pancreatic cancer cell lines, explaining how genetic mutations can activate genetic superenhancers that promote out-of-control pancreatic cancer growth. They show the efficacy of an experimental drug that targets a superenhancer-related protein, demonstrating the promise of therapeutics that block the effects of superenhancers. The pancreatic cancers are among the most aggressive, deadly tumor types, and for years, researchers have struggled to develop effective drugs against the tumors. The researchers have identified a new set of molecules that fuel the growth of tumors in pancreatic ductal adenocarcinoma, the most common type of pancreatic cancer. And that explains how certain gene mutations trigger out-of-control growth in pancreatic cancer by activating a superenhancer that turns on other genes. It also showed the effectiveness of a new drug that put the brakes on pancreatic cancer growth by blocking the effects of that superenhancer. And this is the first time anyone has looked in such detail at the role of superenhancers in pancreatic cancer. The discovery of this superenhancer gives us both basic insight into pancreatic ductal adenocarcinoma, a new way to think of therapies. Enhancers are regions of DNA that, when bound by proteins, boost the expression of genes. This results in higher levels of those genes' protein products. Superenhancers are the most active type of enhancer, and they can be identified by their unique molecular tags. And because superenhancers can simultaneously and powerfully activate many genes, they can rapidly change the state of a cell, switching on cellular programs to encourage rapid growth or change the cell's identity, for instance. In the new work, researchers analyzed 16 different human pancreatic cancer cell lines and identified hundreds of different superenhancers. They pinpointed one associated with the gene HNRNPF that was far more active in pancreatic cancer cells than healthy cells. And then, through a series of experiments, the group showed how HNRNPF sets off a cascade of events, leading to an increase in the amounts of overall proteins cells were producing. It is well established that cancer cells unregulate protein production in order to fuel their rapid growth. They have now identified how cells regulate this process at the superenhancer level. The researchers went on to demonstrate that by deleting the superenhancer or the HNRNPF gene in the cell lines, they can slow the growth of pancreatic cancer cells by more than 80%. They also found experimental drug targeting PRMT1, one of the proteins impacted by HNRMPF activation, to stop the growth of isolated pancreatic tumors in the laboratory and the growth of pancreatic tumors in mice. They were excited to find that there is not a lot of redundancy in the superenhancer pathway, which means there might be multiple ways to impact it with therapeutics. If you disrupt this network at any point, you have the same effect on blocking cellular growth. Additionally, the team discovered that MYC, a cancer-related gene that is mutated in many cancers, including pancreatic cancer, can activate the HNRNPF superenhancer. As drugs effectively blocking MYC have been hard to develop, and the new results suggest a completely different way of stopping the effects of these MYC mutations by targeting the pathway turned on by the superenhancer. And finally, the researchers tested cells from a patient with pancreatic cancer and found that the HNRNPF superenhancer was active in these cells. And these results clearly demonstrate that this superenhancer is relevant in humans and can even be used as a marker to monitor pancreatic cancer progression. More work is needed to discover whether drugs targeting the super enhancer or related molecules might be useful for treating pancreatic cancer in patients. And this is the professor, professor of microbiology, bioinformatics, pathophysiology, and teacher of research. This video was created for educational purposes only. Hope you enjoyed listening. Until next time, have a great day.